So once again, good morning, everyone, viewers at home. My name is Adil Olatunde Michael. I don't want you to forget and lose the focus that we have actually started the journey to designing uh, a vaccine, multi-epitope vaccine against immunogenic genes of multi-drug resistance, Mycobacterium tuberculosis. And for some days now, we have been trying to look at the general workflow if we are to move from wet laboratories to the dry lab. Don't forget, if we conceptualization of that research will actually help us to be able to cover three different three different stages. The first stage is going to be field work. We'll move from the field, and that's why we talk about conducting advocacy, consultation, preparation of this and that, administration of questionnaire, recruitment of our subject uh, into the subject matter, and that is trying to design a multi epitope vaccine against the immunogenic genes of multi-drug resistant Mycobacterium tuberculosis isolates from five southern states in Nigeria, southwestern states in Nigeria. Okay, so we move from the field down to the laboratory, then down again into the dry lab, which has to do with the computational analysis of it. And for some time now, we have been able to dwell too much on all these this. And don't forget that this still remains our focus. Focus where we have okay, this is our workflow. What is actually guiding us? This is it. So we have identification of stakeholders, collection of, and transportation of sample, processing. That is when it has gotten to the laboratory. Laboratory will take the aspects of 3 and 4. 1 and 2 is going to be on the field. 3 and 4 is going to be laboratory based. And then from 5 down to where we are going to talk about later, that is going to be on our computer. So, and that is the computational aspect of it. So we actually started with cleaning the data by the use of FASTA QC in Galaxy. Okay, after that, we talk about streaming and cleaning of this data using FastP, Drimomatic, or what is also known as a cut adaptive. I mean, called adapt. Okay, so in this one, in this typical example, I actually made use of Trimomatic, not FastP. Okay, although FastP appeared to be more recent than any the other two tools. So then thereafter, we are going to make use of a TB profiler. Okay, which was actually installed when we wanted to install Panaroo. Okay, so the genomic assembly was performed using spades. We performed the genomic assembly using spades, whereas the gene annotation was performed using Prokka software. So thereafter, I have installed Panaroo successfully. So the first thing I did was to install Anaconda on my phone. I mean, sorry, on my PC. After that, we installed Ubuntu. So really, I've not actually know much about this Ubuntu, but I know that it is one of the creating the environment of how Panaro is going to work. So Conda was installed, and Panaro was also successfully installed. So the next thing is to actually make sure that we continue from our success of Panaro installation. So, and when you look at this Panaroo, I made a video, I think last night, where I told us about what is a pangenomic analysis, what does it entail, why do we need to, the rationale behind pangenomic analysis, what's the difference between genome and pangenomic. Genome is talking about the same strain, that is with the same organism, but pangenomic is talking about more than one. So, and I told us that that is going to help us, is going to help us to be able to differentiate further annotation of our sequence or our protein. So it will divide it into three. The first one is a core. That one that is if we have four strains, the core is going to be the intersectional space of the four strains. And when you have it in more than one of the strains, you talk about it as an accessory. And if you now have it in only one strain, it will be regarded as just unique, very, very unique. And each of these category of protein, anointed protein, will serve different purpose. Some of them will serve the purpose of whenever we want to just determine the virulence. So we be able to know that, and those ones that will be for vaccine design, we also know that. We also look at what is also called the housekeeping genes. The housekeeping genes are too essential. For example, in cell wall analysis, metabolic proteins, and so on and so forth, replication, the ones that are too essential are called the housekeeping genes. So knowing fully all this, we actually needed to proceed further so that you'll be able to see it live the way we will try to continue to install to carry out Panaroo. And Panaroo, we always take either protein files, protein FASTA files, or GFF3 files. We will know more about this whenever we go on. And just like I said, Pangenome with Panaroo actually explain that it will identify different core genes, accessory genes, and the one that are unique. And it's going to tell us those ones that can actually be used as biomarkers for vaccine development. So these are core proteins. Not only that, they are also conserved. That is, you can find them in not only one strain of Mycobacterium tuberculosis. So having known that, so we cannot successfully carry out this without having more than just one strain of Mycobacterium. Okay, so and don't forget that we already have in this work specifically, we already have samples isolate from four different states. That is Oyo, Ondo, not only Ondo, Ekiti, Kwara. So at least we have isolates from these four uh, states. So that is to tell you that that is the reason why we need to TB profile these isolates before we'll be able to know how to carry out make some inferences so about that. The TB profile is going to really assist us to be able to profile to know the genealogy or the lineage of our strain. So then it's also going to help us to be able to identify drug resistance genes as well as those ones that are mutation that have been changed. That is the mutational genes. Okay, then so it will also help us to be able to predict the antimicrobial resistance. So for some of us that are in antimicrobial drug resistance, that means you will need, although all of us, all the three groups, that is the vaccine, the drug, as well as the antimicrobial group, you are going to need TB profiler so as to give you the idea of how your microorganism, that is Mycobacterium tuberculosis, will always look like. So apart from that, we will now So we will now try as much as possible, because what we have been dealing with, the isolate we have been dealing with, as you know, in Galaxy, that we have been running all this while, it was just an isolate from Quara, that is from one side. That is, we have only one strain. So that is telling us that we need to get more strain. So that is going to be the next task for us now. The next task will just be that I will start all over again from genomic analysis, whereby I'm going to quality control clean using Galaxy, fast QC. So I'm going to subject all the other three into um, into the other aspect. So what I'm saying in essence is just that normally some of us were given only one sequence. So that is to tell you that you need to start again so that you'll be able to run Panaroo, because Panaroo is only being can only be run. That's why they call it Pangenome. So we want to identify those genes that are housekeeping, those genes that are too essential, those genes that can be responsible for virulence, for antimicrobial resistance. All these are essential genes. And that is the reason why any reasonable intervention will focus on any of these category. Any intervention that is not targeting any of these housekeeping genes might eventually not be able to be of any essence at the end of the day. So that's the reason why I want to start again. I will start from QC, trim, and clean the data by removing the adapter. Then I will now run the TB profile of all the four together. So after doing that, then we'll now do genomic assembly, gene annotation using Prokka, so that by the time we now get it done that same way, we can proceed from there by running the Panaroo, that is the Pangenomic Analysis, from there using Panaroo. Okay, and after doing that, we'll now go into we'll look at how it is going to be. These four will look like under BVBRC platform. Then before we will enter into immunoinformatics, that is the reverse vaccinology aspect. That is, for those of us that will be interested in designing vaccine, we will do that. And for some of us that will be, our interest is on drug, we are going to focus that. And for some of us that will actually need to run other antimicrobial resistance database like CAD, like REST finders, and some other database, uh, we will be able, like the use of Patrick, we will use Patrick as well. So that we'll be able to do it once and at a time. So without wasting our time, let's go back to our galaxy. Let's go back to galaxy. Let me start it again. Let me cancel so that you come to tools. Come and click on tools. Click on tools. So inside the search box of tools in Galaxy, you are going to type fast QC. That is, you want to quality clean your data. Okay. So having done that, you click on multiple data sets. Okay. Then here you click on upload. Okay. Click on upload here. So when you get there, choose local file so that it's going to lead you to where you have your TB sequences. Don't forget that I told you, just like as I told you that the this time around we are going to run more than one string together. So this is my own my six TB sequences. I have them here. So what we will do, so once again, it's better let us just do um, all of them at a time, all the six at a time. Okay, this is the reverse, this is the forward, reverse, forward, reverse, forward. So then just click enter so as to upload. So these are your 1, 2, 3, 4, 5, 6. So these are your four different. It is expected that because the location are four different locations, so it is expected that we are going to have three. Okay, it is three. It is three. One is still pending. One is still pending, and that is the one from Ekiti, and the other one. I have two isolates that are still left somewhere, okay, which we do not have the because of it did not pass through the quality control level, and that's why that one has to be discarded. I will now look for another very clean isolate again that we are going to be able to sequence them. So after this, I have two more sequences that we later on. But let's not just run for these three first. So by the time we run for these three, let's see how it is going to be. So under normal circumstance, that means we are going to be dealing with three different strains. Okay, so you can see these three, this is don't forget, wherever you see one, that is forward. Wherever you see two, that is a reverse. Forward, reverse, forward, reverse. So you now upload them. After you must have uploaded them, just click on start. By the time it starts, just be watching here. You will see that it is going to um uh what is the problem? Okay. This is not the galaxy I was supposed to use. This is not the galaxy. This is the galaxy I was supposed to use. So that one I've not actually registered under it. So let me just, in order not to be confused, in order not to be confused, anyway, let me upload it again. Fast QC, fast QC, okay, read quality, this. So you can see because this is already a registered galaxy. So that's why you can see my name there. I do it Olatunde Michael. So this other one, if you look at it, it has no history, and this one is requesting that I should log in or register. So that is to know. So anytime you want to any work you do here, it might not actually be too safe for you to do it there. Okay, so that is that. Let me fast QC, as I said, uh, tools, tools. So fast QC, QC, read quality control. Okay. So instead of multiple data sets, you use okay. So I think the best thing is to delete. Please let us delete everything here so that it we won't be able to. It will not. There won't be any mixed up at the end of the day. So check again. This is Adeoto Olatunde. Okay. So fast QC, always check this. And this one is under tools. Tools, fast QC. Okay. After that, click on multiple data sets, then upload. Okay. File, choose local file. Then these are the sequences. 6. Okay. 1 2 1 2 1 2. Okay. So let us now start to run this. So as soon as this is started, you will see that you see progress here. And under normal circumstance, at a point in time, very soon, we are going to have it being run here. Then please wait 6 out of 6 remaining. So this has actually confirmed that this is what. So I want you to start doing something so that you won't need to wait so many times, so many precious time again. Now that we are running this, I want you to screenshot this. Just screenshots, okay, so that you'll be able to keep. So I have screenshot this, okay, and I'm going to keep it somewhere later. So let me try and save. So this is from fast QC, fast QC progress, in fast QC. So I have it. I'll be storing all this in my. Should I be stored? Okay. So I think this has been stored. So you can see the progress. So immediately it is completed, it will move to this panel. It will move as soon as it reaches 100%. It's going to move to this place. So that's why you need to monitor the progress here. Okay. Let us wait. This is going to take some time, and it might take as long as 1 2 hours, depending on the available internet space. So please let it go. Um, let's wait and see the progress. So, but because this is a video, what I will do is maybe later I might need to edit. I don't know, I might need to edit, but if I cannot edit, just to make sure you have you have idea of what is going on. Don't forget that this is live. This is live streaming. So let's monitor how it's going to be in the next few hours. Yeah, thank you. So let us try as much. I want to still engage you so that it won't be just too boring. Let me go to Zuno so that I will be able to start giving you music, that is, so that I will be able to give you musical interlude so as to enjoy your day. Okay. So we wait for Zuno.ai. So, and I'm going to go straight into the library, my library, musical library, and I'll start playing so that you enjoy the rest of your day here as we monitor the progress from
Ba 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 da. Ba 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 da. What are the outlets in the medical meat? Tiny track with pulses in tech, which is warm strings and big passion. Futurist science mood representing the shift from wet labs to dry light. High energy breakthrough tone, no lyrics, only your pets. Ba 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 da. 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 Drowning it, ever cinematic. Hologram track with pulsing synth. Tech glitches, warm strings, and big brute quiet. Futurist science move representing the shift from wet labs. Try out with high-end breakthrough tone. No lyrics, only vocal pads. Da na da 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 da. Da 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 da. Da 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 da. 전화를 하고 dynamic electronic jack with cousin sins. Double bridges, one-sys, umbrito cara. Future insights to the prison. The shift from wet labs to dry lab. Giant boats, high energy, breakthrough tunnel lead. Only Coco pass. 좀 나니까 alligator 나락이긴 레전이 trapito swings to tapito one strings and big percussion futuristic science mood representing the shift from wet labs to dry labs.
High energy breakthrough tone.
No lyrics, only pass. Futuristic cinematic science track with true elements. Glowing digital and shows you subtle labs. Sound effects, animatronic and SI mood represents journey from wet labs to dry labs. Intelligent, modern, motivating, light on sway, vocal tricks days. Futurism, cinematic science track with on-field elements. Glowing digital applications, subtle lab sound effects. And it's your rocking line moon represents journey from wet labs to dry labs. Intelligent, modern, innovating, light at its fruit. Blah, 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 blah. Lighter than my love. Blah, 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 blah. Lighter than my love. Blah, 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 blah. Lighter than my love. Futuristic and science track with simple elements. Glowing digital robots, fate turns out in man. Side effects, energetic, lab like Citrus labs and labs and living more than and that's a punk Futuristic symbol like science track with simple elements. Glowing digital application satellite. Sound effects, sound effects, sound effects. Represent journey from wet labs to dry labs. Intelligent, guiding, light on me like X. Python for everyone. Python for everyone. Python for everyone, Python for everyone, Python for everyone. That brings and is persons, shall find harmony that brings and is persons, shall find harmony that brings and is persons, shall find harmony that brings and is persons, and is persons. Python for everyone, Python for everyone, everyone. been the greatest. You see me winning, you see me slaying, you see me grinding, you see me hating, you see me saving, you see me praying for the Lord to come and help me with the fading. Yeah, I've been the greatest. You see me winning, you see me slaying, you see me grinding, you see me hating, you see me saving, you see me praying for the Lord to come and help me with the fading. Yeah. I've been the greatest, you see me winning, you see me slaying, you see me grinding, you see me hating, you see me saving, you see me praying for the Lord to come and help me with the fading. Yeah. I've been the greatest. You see me winning, you see me slaying, you see me grinding, you see me hating, you see me saving, you see me praying for the Lord to come and help me with the fading. Yeah. I've been the greatest. You see me winning, you see me slaying, you see me grinding, you see me hating, you see me saving, you see me praying for the Lord to come and help me with the fading. Yeah. Yeah. I've been the greatest. You see me winning, you see me slaying, you see me grinding, you see me hating, you see me saving, you see me praying for the Lord to come and help me with the fading.
been the greatest. You see me winning, you see me slaying, you see me grinding, you see me hating, you see me saving, you see me praying for the Lord to come and help me with the fading. Yeah, I've been the greatest. You see me winning, you see me slaying, you see me grinding, you see me hating, you see me saving, you see me praying for the Lord to come and help me with the fading. Yeah. I've been the greatest. You see me winning, you see me slaying, you see me grinding, you see me hating, you see me saving, you see me praying for the Lord to come and help me with the fading. Yeah. I've been the greatest. You see me winning, you see me slaying, you see me grinding, you see me hating, you see me saving, you see me praying for the Lord to come and help me with the fading. Yeah. I've been the greatest. You see me winning, you see me slaying, you see me grinding, you see me hating, you see me saving, you see me praying for the Lord to come and help me with the fading. Yeah. Yeah. I've been the greatest. You see me winning, you see me slaying, you see me grinding, you see me hating, you see me saving, you see me praying for the Lord to come and help me with the fading. Go on and start a war. Whatever makes you smile, start a war. We can start a war. Come on. Whatever makes you smile, start a war. Forget about all the 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 war. Start a war. Whatever makes you smile, start a war. We can start a war. Come on. Whatever makes you smile, start a war. Forget about all the 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 war. Go on and start a war. Whatever makes you smile, start a war. Go on and start a war. Whatever makes you smile, start a war. We can start a war. Come on. whatever makes you smile. Start a war. Forget about all the 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 war. Go on and start a war. Whatever makes you smile, start a war. We can start a war. Come on, whatever makes you smile. Start a war. Forget about all the 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 war. Get your hands up. I need y'all to be activated. I need y'all to be motivated. I need y'all to be educated. I need y'all to be excited. This is our year. This is our time. This is our moment. That's what we're made for. We were made for such a time as this. This is what we're here for. And I'm here for you. And I'm here with you. Yeah. Let's go. Let's go. We in the house tonight, let's go. This is a celebration. We celebrating us tonight. We celebrating us tonight. We in the house tonight. We in the house tonight. And we're here to celebrate us tonight. We're here to celebrate us tonight. So let's go. Yeah. Let's go. We're in the house tonight, let's go. We're in the house tonight, let's go. Let's go. Let's go. Let's go. We in the house tonight, let's go. We in the house tonight. Let's go. Let's go. Let's go. Let's go. Let's go. Let's go. We in the house tonight, let's go. We in the house tonight, let's go. Yeah, let's go, let's go, let's go. We in the house tonight, let's go. We in the house tonight, let's go. Let's go, let's go. We in the house tonight, let's go. We in the house tonight, let's go. We in the house tonight. We in the house tonight. We in the house tonight, let's go. Let's go. Let's go. We in the house tonight, let's go. We in the house tonight. Let's go. We in the house tonight, let's go. Let's go. Get your ass up. I need y'all to be activated. I need y'all to be motivated. I need y'all to be educated. I need y'all to be excited. This is our year, this is our time, this is our moment. That's what we're made for. We were made for such a time as this. This is what we're here for, and I'm here for you, and I'm here with you. Yeah, let's go. Let's go. We're in the house tonight, let's go. This is a celebration. We're celebrating us tonight. We're celebrating us tonight. We in the house tonight, we in the house tonight. And we're here to celebrate us tonight. We're here to celebrate us tonight. So let's go. Yeah, let's go. We in the house tonight. Let's go. We're in the house tonight. Let's go. Let's go. Let's go. We're in the house tonight. Let's go. We're in the house tonight. Let's go. Let's go. Let's go. We're in the house tonight. Let's go. We're in the house tonight. Let's go. Haven't I given enough? Given enough? Haven't I given enough? Given enough? Haven't I given enough? Given enough? Always the fool with the slow hand, looking for answers in the pouring rain. You wanna find peace, love? It's not hard when you wanna be heard in the wrong ways. 'Cause I'm in the hardest on you. I said 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 I'm in the hardest on you. Haven't I given enough? Given enough? Haven't I given enough? Given enough? Haven't I given enough? Given enough? Always the knot in the wrong hands. I carry guilt like a ball and chain. You wouldn't apologize, no. It's not hard, it's too late. I can't undo that. 'Cause you took the hardest fall. Oh, I said 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 you took the hardest fall. Oh, 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 oh. I said I've been the hardest on you. Oh, I said I've been the hardest on you. Oh.
So, yeah, welcome back. Welcome back. I want to really appreciate you for staying online up to now. I will soon be going off the line. People that know should know the reason why. Let me go and watch my football club, Manchester United. So we are going to return once again. Let me just appreciate you. I'm going to return later in the day, where I'm expected to resume from where we are stopping now. Don't forget that what we have done so far now is to still go, and in order to be able to meet up with the condition or the conditionality of the pangenome that has to do with more than one strain together, the genome of more than one strain. That's what we want to compare, so as to be able to identify the core genes, the accessory and the unique genes among and between different strains. Don't forget that this work actually started with three, three strain, at least three strain, which I told you two will still be coming, still under processing. So when we come back, I'm going to continue from where we stop and where we are stopping, and that's going to be is either we use fast P or Thermomatic in order to be able to trim and clean our data. Thereafter, I'm going to subject my data to TB profiler, and I'm going to make a special video on TB profiler because that is another very important tool, bioinformatics tool that we make use of whenever we were trying to do the genomic analysis of it. So it's very, very important because that we are going to know the antimicrobial resistance from there. We are going to know a lot of things about that. So other information that we will need, I'm going to let you know. Then we'll come back, run this genomic assembly, do the gene annotation.